So today we're going to talk about chapter 3, which is all about development of the brain and also how hormones factor into that. More specifically, we're going to talk about the development of the nervous system from, sort of the creation of a cell all the way to what we see as maturing brain development in utero and after birth. And we're going to talk about those stages of brain development. We're also going to touch on hormones. We're not going to get into this in great detail, but we're going to talk about the endocrine system, how hormones are secreted, and types of hormones. But we're not going to get into super detail about the pituitary gland and the types of triggering processes. We're just going to sort of touch on that based on how it relates specifically to development. So looking at the development of the nervous system in utero, it happens really early in gestation to formulate what becomes the brain and spinal cord. And what you'll need to know in this process is how it starts in terms of just the cellular process. So here, in this first sort of picture right here, you see that it starts as something called a neural plate. So this is an example of a 20-day-old embryo. And so here you can see that sort of the base right here, right? Okay, you can see that it actually forms a plate of cells. It's kind of like a flat plate. And as the cells start to build, they actually start to kind of come up into a groove, and that groove looks kind of like this as it starts to form. Here you can see this is kind of the next sort of step. And then as it moves into just day by day, you can see here this is a 21-day-old embryo. It starts to move from this plate to a fold to a groove as it starts to fold in on itself here. And then by 24 days, you see that, so here you can actually see it goes from a plate to more of a fold to a groove here. And these cells kind of fold in on themselves to secure what we refer to as the tube, the neural tube. So here's a 24-day-old embryo. Here we have the tube secured and folded over. And now this is going to be the preliminary brain, okay? And what's really important here is that we need this tube to close. So here you can see that as the tube has closed, now we're starting to form the spinal cord. And if you don't have the full closing happen, you can experience this condition called a spina bifida. And spina bifida can occur in varying degrees of severity. So you can have not completely closed spine or tube here, and then that can lead to a more mild version of spina bifida, all the way to a really severe form where the fetus would not be able to survive, or the embryo wouldn't survive. And then sometimes you do have cases where a person is actually born with spina bifida with more severe cases and would need surgery. There are tests done with a pregnant mother to determine the risk of spina bifida, and if that is something that they have to kind of keep in mind if they want to continue the pregnancy. But here, as you can see, that the matter here is made up of white matter here, and then the spinal cord this part is made of the gray matter. So another kind of view of this is looking at how the brain is forming really early on in gestation and how that builds up to what we see as a mature brain. So this is really nicely color-coded, so you can kind of see here that the forebrain is in this color here, which matches the color here, this forebrain. And you can see as it's forming into the telencephalon and this structure here, which is the forebrain. The midbrain is in this kind of purplish color, and as you can see here, is forming into the mesencephalon, which you can see here. And then here, the hindbrain structures are dividing up into the myelencephalon and the metacephalon here, which are located here. And so, again, you can see even more structures specifically where these structures are developing in utero, and what sections of the brain that early development then ends up developing into. And we're going to talk even more specifically about how this kind of works in a really interesting graphic that shows you how the midbrain sort of kind of goes up into what we see as sort of covered by the cortex. So we're gonna cover that as well. But it's really important to kind of see how it starts out with the forebrain, midbrain, and hindbrain, and then how those structures then come to be in terms of the course of development. So if we start with the forebrain contains the lateral and third ventricles. We also see that it's divided in the telencephalon and the diencephalon. The telencephalon is made up of the cerebral cortex, basal ganglia, and limbic system, whereas the diencephalon is made up of the thalamus and hypothalamus. The midbrain contains the cerebral aqueduct, which is really important for moving the cerebral spinal fluid through the brain. Here, the mesencephalon is made up of the tectum and the tegmentum. The hindbrain, of course, contains the fourth ventricle, made up of the metencephalon and myelencephalon. The metencephalon is made up of the cerebellum and pons, whereas the myelencephalon is made up of the medulla oblongata. And here we can see even further how the course of development occurs. So here you can see starting at 3 weeks, it doesn't look like a distinguishable brain, but the process is occurring. Here we can see 25 days, just like as we talked about earlier, we see that forebrain, the midbrain, and hindbrain very clearly starting to develop into the structures needed to then migrate to the places that they end up being in the brain. So here you can see in 4 weeks to 5 weeks, moving on to looking into more of a distinguished brain. It's not really until 11 weeks that really we start to see that brain start to take more shape. You can see here that the structures aren't where they need to be, but you can see it start looking more like a human brain. Here you can see 35 days, the embryo is 45 days old, and here the structures are moving into where they're going to be. Again, here we see that movement still occurring. By 50 days, you're starting to see this structure here starting to look more like a forebrain. But again, the midbrain is still exposed, not where it needs to be. We start to see, and then here you can see actually the spinal cord, which is nice. You can see where that part of the brain here is sort of starting to be where it needs to be. By 100 days old, that embryo starts to look, or at this point the fetus is starting to look quite like a brain. The midbrain here is not where it needs to be, but it's moving up. It's moving up into the forebrain area and being covered. Here the pons and medulla, which are really important for survival. This area here is sort of what we call the older brain, important for things like breathing, heart rate, important vital functions. And then here by 5 months in utero, you can see that this fetus has a brain that looks quite like a more mature brain. The midbrain is now from here covered by the forebrain. And then here you can see the hindbrain where it needs to be. Still some areas that are exposed here, but by 9 months, you're starting to see a much more mature looking brain that looks like what it should for developmental purposes. And then we, of course, see it because here's the cerebellum. So the only part of the hindbrain that's exposed here is really this portion. And then, of course, the pons and stuff are located down here. And the midbrain is hidden up into those structures. So as development occurs, we're going to talk about how specifically these cells are generating, how they're migrating, differentiating, all that as the process occurs. So here are the different stages of brain development and we're going to go through each one of these individually, going from cell birth or neurogenesis, it's a good kind of tag word to remembering sort of birth. Migration, differentiating those cells, the cells maturing, synaptogenesis, creating new synapses and new connections. We're also going to talk about neuronal death, which is also important in the process, as well as myelogenesis, which is creating new myelin sheath and coverings there. So we're going to go through each one of these so you guys are familiar with the different terms and then how it specifically occurs in the course of development. So cell birth or neurogenesis, this is the generation of new neurons. And in utero, we're seeing this process occur with neural stem cells. So neural stem cells are those that can really be any kind of cell in any location they want. And as they are, sort of as we're birthing new cells, we see this occurring in the neural tube during development. It's going to continue that course of creating new cells throughout the brain. For a long time, we thought that this process only occurs in utero and shortly after birth. And then once the cells are generated, that's it, we're done. But as we are always learning new things about the brain, relatively recently, we discovered that there is neurogenesis occurring in the adult brain. And so this started actually in some rodent research that found that the area of the hippocampus, so specifically the hippocampus, which is important for generating new memories, we're seeing that neurogenesis is still occurring. So we're actually creating new neurons in the hippocampus even as adults. But we don't have very good evidence about this occurring in many other areas of the brain. We're still learning about that process. Another really important process that occurs in this stage of development, so we have cell birth and creation of new cells, is cell migration. So this is the cortex. This is an example of layers of the cortex here. And so what's important here is to understand how the cells build on each other to build the layers of cortex. So in an adult brain, we have 6 layers of cortex. And what's responsible for this process is radial glial cells. So here we have this radial glial cell here, which is guiding the neuron. So here we have a neuron, guiding it to its correct place in the layers of where it belongs. And so because we see, for example, okay, this one's guiding, we have to layer 1, the next layer is going to be level 2, the next layer is going to be level 3, and building those cells at each layer. And because it goes from, starting this and then building on top of it, it actually is developing the cortex layers inside out. So the last layer to be formed is layer 6. Remember, we have 6 layers of cortex. A very important number to remember. But as these layers are sort of building on themselves, okay, that's going to really finish this final layer at layer 6. Now, this process of developing layers is actually, it's not a short, fast process. This is a pretty long process, and it takes about even 8 months after birth to finish. So this is a process the brain is still developing. It's still, moving those cell types. It's still making sure, solidifying those layers and creating more synapses, and which we'll talk in a minute. All of those things are still occurring even well after birth. So this process, pretty much, is layered up by about 8 months after birth. So looking at some other stages of development, of course, we have cell differentiation, which is going to have that cell type really differentiate into exactly what kind of cell it's going to serve as. Is it going to be, a motor? Is it going to be a sensory cell, a motor cell? Is it going to be, where is it going to be located, and what is its function going to be? Cell maturation is also important in this stage of development, which is a two sort of pronged process. We have a sort of a fast growing process and a slow growing process. The slow growing process is growing those dendrites, making sure that we're increasing the surface area of the cell to allow to provide an area for synapses and other cells to network and connect with each other. The second, more quickly growing part of this is cell maturation, is extending those axons to appropriate targets. Now, as you are navigating your environment as an infant and being exposed to more things and learning, this process can grow. This process can happen really quickly. You're constantly sending out these axons to different targets. In order to, again, establish connections and be able to network those cells better. Another process that occurs is the synapse formation and synapse pruning. So synapse formation is creating new connections, new neuronal connections. Extending out those cells, guiding the axons, guiding those dendrites, forming connections from cell to cell. This occurs throughout your life. And as you continue to learn and create new experiences, those synapses in your brain are going to continue to do this process of creating new connections. Now, on the other side of this, we also have something called pruning. So pruning is just like you would expect to do to an overgrown bush in your house, outside your house. And that is to kind of, if you're not using certain connections, they prune off. They just don't connect anymore, and they'll shrink back. And so this is not necessarily a bad thing. So people say, oh, my connections are pruning. Well, this is good, because if you're not using those network connections, then that allows growth and maturation of other network projections that you're actually using. There's only so much space in the brain. We only have so many cells. And so, of course, this process is really important for just being able to manage the kinds of cells that we have in the brain. So, pruning is not necessarily a bad thing, but it is something that can be a bad thing, obviously, if it happens in excess. But this is just something that our brains do regularly all the time. Now, a little bit more about the neuronal cell death and synaptic pruning. So cell death is referred to as apoptosis. So apoptosis is, this is something that, again, is important in the process of development. And that is because in order for, for example, a fetus or an infant to create new cells and to create new connections, sometimes those cells, live their life and they die in order to allow for creation of other cells. Now, if you interfere with apoptosis and you don't let the cells run their life cycle, you end up having a brain that ends up being too large to fit in the skull. And so, of course, we want that would be dangerous for the survival of the species. So cell death is part of the process that happens. And so after normal cell death, of course, those synapses are going to rearrange themselves and develop those appropriate connections. And we're going to see this process occurring throughout a person's life. Cells have a lifespan, and those cells need to die when they are sort of aged and ready to do so. Now, what we also find is that synaptic pruning does have a pattern. And so the last place that is in the brain that matures is the frontal cortex. Okay, so this sort of area here is the last place that really finishes maturing, both in terms of pruning as well as in myelogenesis, so forming myelin. And both processes occurs from back to front, meaning that it starts here and works its way to the front of the brain. And the prefrontal cortex area is last to be myelinated, also last to be mature and completely developed. And so, of course, there's debate about exactly how old you are when your brain is finally mature, and most settle around about 25 years old as having the final sort of stages of maturation where it reaches the prefrontal cortex, which is why there's arguments about, should we have people making adult decisions if their brains aren't actually completely mature? But that's sort of an ethical debate, and some people say, well, I'm 18 and I can make decisions and I think I'm an adult. Whereas the brain development patterns suggest that really it's about 25 years old where we see the final maturation happening. And so during this process, of course, of brain maturing, and it sort of following this pruning pattern, we actually do see some areas of development, some timing of development where the pruning occurs more often than not. And so one stage of development that we see is puberty, where we see pruning occurring right before the puberty hits. And so like I was saying, it's really important that this pruning process occurs, because we know that there are associations between gray and white matter, and how much that matter that you have, and how you had these abilities sort of your cognitive abilities are associated with this. So specifically talking about cortical gray matter. So remember, white matter is the axons. It's the the myelin sheath that make up the white matter. The gray matter are like the dendrites and connections. What's interesting is that we need to have these pruning. We need this to happen in the stage of development to have a healthy brain. And so we find that there needs to be a reduction in gray matter in the primary sensory areas, as well as the secondary regions during this pruning process, to have a healthily developed brain. We actually do see, in cases of looking at sort of correlational studies, that cortical gray matter, the density, so how much you have, how dense it is actually negatively correlated with cognitive abilities. So what is a negative correlation? That means that the more gray matter you have, okay, so the more gray matter you have, the less your cognitive abilities. And so, of course, if we reverse that, then that's saying that those with less density, less dense gray matter, tend to have more cognitive abilities. Of course, there is a line here, right? So, I mean, if you have very, very sparse connections, that's a problem. But if you have too many, that's a problem. And so we don't want the brain to have too many connections, because that is actually linked to issues with having a healthily performing brain. So let's cover hormones. So we're going to talk about hormones specifically and how they relate to development. So there's only a few slides on this, and I'm just covering kind of the basics on this. We're going to talk mostly about the endocrine system and specifically the endocrine glands. So endocrine, meaning within, crinin meaning secrete. This is important for secreting hormones. We're going to talk about the difference between hormones and neurotransmitters here in just a few minutes. Endocrine glands are the ones that usually are the ones secreting those hormones and also producing them specifically. And so the endocrine glands are going to release those hormones in the bloodstream and within the body to send messages that are in some cases permanently changing the body, and in some case non-permanently changing the body. We're not going to really get into this, the exocrine system or glands, which is secreting fluid outside of the body, exo meaning outside, right? Endo meaning inside. So exocrine system is all about your sweat, your tear glands, and your salivary glands. And we're not going to really get too much into that. This is just letting you know that there are two different systems. What we will talk about, though, is the difference between neural and hormonal communication and what makes them different. So looking at the kind of levels here. So what is the type of message? How fast does it transmit the message? What type of signal? Where is it going? And how is it traveling? These are the distinct 5 sort of characteristics that we look in distinguishing a neuron's neurotransmitters and an endocrine glands hormones. So neurons are which release neurotransmitters. Okay, these are traveling over fixed pathways. They're sent to reach a very specific destination, whereas hormones tend to spread throughout the body, throughout the bloodstream, and they're picked up by cells with receptor types specific to that hormone. Neurotransmitters are released very, very rapidly in a matter of milliseconds, whereas hormonal release tends to be much slower. You can actually measure it more in seconds and minutes because it's a much slower process. The signal of neurons releasing neurotransmitters is all or none. It's an action potential that either fires or it doesn't. There is no partial signal that's sent. It's an electrical impulse that's sent, and those chemicals are then being released. Whereas with hormones, it's a graded signal. That means that as it gets closer to its destination, it gets further away from the source, you're actually seeing that the signal tends to be graded, taking over especially longer distances. You're not getting the same signal at the point of contact as the receiving end. Destination for neurotransmitters is very, very precise. It's the postsynaptic cell for those connections, whereas for hormones, it's all throughout the body. We're talking, getting picked up by specific receptors and usually going through the bloodstream. And for neurons, the distance between neuron to neuron is extremely short, and talking nanometers, whereas with hormones, they're traveling over a much larger distance, in some cases as far as over a meter, from point of release to point of reception. Just really sort of briefly, we're going to talk about how the hormones are triggered and secreted. So the hypothalamus and pituitary gland, really important for sending and receiving. The pituitary gland is sort of split into two, but we're not going to go into the anterior and posterior. We're just going to talk about it sort of as a whole. The hypothalamus, so the pituitary gland's here. The hypothalamus is made up of a series of nuclei. A lot of people don't realize this. They think that the hypothalamus is just like one structure, but it's made up of a bunch of different nuclei. Like, for example, we'll talk about the suprachiasmatic nucleus when we get to sleep and circadian rhythm and how that's regulated. But these different nuclei have different jobs for different kinds of specific hormones and neurotransmitters release. Specifically neurotransmitters. Just a reminder that proteins are synthesized within all cells, and some are specifically used for communication between cells. We're going to talk about hormones, neurotransmitters, and neurohormones. So hormones, of course, are produced by the endocrine cells, and they're released into the circulatory system and into the blood. Neurotransmitters produced by neurons and then released at the synapse, presynaptic post, and then it's going to send to the postsynaptic area in the synaptic cleft. And then here we have neurohormones, and these are produced by specialized hormones called neurosecretory cells. And we've, and these are just, again, this is just the basics, these are the kinds of communication that's sent through. Now, of course, as we talked about in this chart here, okay, the main differences between those types of chemical communications: where are they produced, where are they released, and what is the distance traveled to produce that effect. And we talked a little bit about that in that table prior to this slide. The role of hormones in development, they serve essentially two major effects, and then we're going to talk about these just sort of briefly, specifically in gonadal hormones. So first of all, hormones can act to organize development. So they act in an organizational way, and they organize development of the brain and body in a permanent way. So for example, testosterone is released during puberty and is going to affect the male body, specifically causing hair growth, a deeper voice, things like that. So these organizational effects are permanent. They're going to change the body in a way that is really not easily reversible. And we're not going to talk about things like transgender and being able to potentially reverse some of these processes. Some of these processes still can't be really reversed too much once these hormones have done their action. That is kind of set in place. There's some ways to kind of work with that and tweak that a bit, but specifically these are permanent actions. Then, of course, the second type of effect is activational, which is hormones are activating certain phenomenon, and these are not permanent. So, for example, luteinizing hormone is going to trigger ovulation. That happens, every 28 days or so many days, every month or so, in a female body. And that is not permanent. It's going to happen sort of like clockwork, but it's going to activate a particular process. Now, specifically with gonadal hormones, and so gonadal hormones specifically in testes and ovaries, we know that androgens, for example, play a really, really big role in sexual differentiation. So estrogen's important, but the androgens seem to be playing a bigger role in the brain of primates. And we also know that hormones are really important for those organizing effects to differentiate a male and female sex. So if you look, for example, in utero and that critical timing, we're seeing that the penis is being formed versus the vagina, and so that's happening in utero, and the androgens are really important for that. For example, testosterone, very important for that process, and we're also seeing that it activates other sex-specific physiology and behavior later in life. Puberty being something that is that's triggered by hormones to set off a cascade of organizing effects that we see in a human body. Now, we know that gonadal hormones are not only important for critical periods of development, but they're also important throughout life in terms of what you see in terms of brain development. So gonadal hormones are important for lots of stages of cell development, going from neurogenesis all the way to cell death, axon guidance, synaptogenesis, all those very important stages. It's really important for those critical periods and for timing. For example, for puberty, that's all timing specific to what's going on in the body. We know also that estrogens are really important for protecting against cognitive decline, and both estrogens and androgens being, the two primary gonadal hormones are also important for playing a role in myelination of axons. And so this final slide is just some of the glands, specifically where these different endocrine glands are doing this function within the associated hormone. So this is just kind of an overview. We talked about the ovary and testes with estrogens and androgens, and specific types of each are located here. But this is just kind of an overview of some of the things that we see that are really important for development as well as throughout the lifespan. So if you have any questions, you can email me specifically about this, and I will be happy to answer those.